Hello everyone, my name is Kari Sogera Iamba and I will be presenting about our experiments in Papua New Guinea. We studied the role of insects and non-woody plants in tropical forest succession. It is a community-wide experiment that was replicated at the lower and higher elevation. Successional changes are primary mechanism of ecosystem regeneration and herbaceous plants are known to block succession for the forest to reach like woody vegetation. And herbivorous insects have known to promote diversity in tropical forest systems, and they exert a top-down control and they promote plant diversity in the primary and also in secondary forest. The recruitment process during secondary succession also opens up the forest systems for invasion of the plant community by alien plants, which are more demanding, which demands more nutrients from the soil and also light from the sun. Our experiment mostly concerns the hilly part of the succession, and that is from after clearance, where we have the herbs and the grasses, to hilly successional vegetation, where we have woody plants and also we have the shrubs. So this is an example of our experiment in Papua New Guinea. So after the soil is been cleared, uh, it's been taken over by this. We can see that there's some herbaceous plants growing, and then from there they develop into shrubs. There's recruitment of shrubs, and then eventually to small trees or pioneer woody plants. So we wanted to test the impact of herbaceous plants on early secondary succession, and also to test the impact of insects. We hypothesized that non-woody plants can block the vegetation to reach their woody component, and uh, we also hypothesized that they will also slow down diversity, and this effect will be more stronger on the lower and weaker at the higher elevation. Insects have known to reduce biomass and also increase species richness, but again we predict that effect to be more stronger on the lower and weaker at the higher elevation. We also predict that insects will mediate the interaction between non-woody and woody by preferentially feeding on the non-woody and thus facilitating the succession towards the woody plants. And this effect will also be stronger on the lower and then weaker at the upper elevation. And also alien plants, which are known to escape herbivory, will be more prominent at the lower than at high elevation. Our experiment was carried out at two locations. The first one was at Numba, which is about 700 meters above sea level, and the second one was at Hubi, which is 1,700 meters. These two location, it's important and it's interesting because Ubi at 1,700 meter is located in a very terraneous area with lots of mountainous area in the Finisterre Mountains, and then we have Numba, which is located along the central Cordillera. So the Finisterre range is a smaller replica of the bigger central Cordillera, and they have ranges from 500 from 500 or 200 meters all the way to over 3,000 meters above sea level. So our two sites was separated by 1,000 meter. So it is an altitudinal difference of 1,000 meters. We had four treatments. in treatment one, we used as it is we used it as control, so there were no application treatments. In the second treatment, only insecticide was applied. In the third, we remove the non-woody plants, the grasses and the herbs. And in the fourth, we remove the herbaceous plants, but we also applied insecticides. And we had 9 blocks, 9 replicates at each location at the lower and higher elevation. Uh, pioneer species have very small seeds. They're numerous and they survive long in the soil. Mostly the non-woody plants, herbaceous and grasses. Whereas the primary forest species like trees, woody plants, and the shrubs, mostly the trees, they have large seeds and they have low germination rates. They survive long as seedlings. And in order to ensure that these different mixes of pioneer non-woody and woody plants have chances to germinate, we mix the soil. So the soil bank or the seed bank was mixed. So we remove all the soil from the 5 by 5 plots to a central mixing area. As you can see, the blue canvas, the soil was thoroughly mixed and then redistributed back randomly to the plots. We applied 3 types of insecticide. First one is chloroquine, which was applied every 2 weeks, and that focused on suppressing the root herbivores or the root insects. Lambda was sprayed every 1 week, and it was fast action. It was like applied on the foliar, and then it repels herbivores from feeding on the foliar. Conpita was applied every 1 week, and it's as a systematic uh, mechanism which is uptaken by the plants and prevents uh, insects or herbivores from feeding on the leaves. For the weeding treatment, we remove both the native and the alien. Some of the alien plants, Paspalum conjugatum, which is a grass. We also have a native grass, which is Ceteria palmifolia, and we have the alien herbs, the Ageratum conicoides and Elephantopus mollis. And on the top right, we have an aerial photo of the 4 plots. And then at the top, below the right top, we have completed or finished established plots after the soil mixing has been done. We also collected soil samples, and this is still pending nutrient analysis, and this will be used to measure soil nutrients like carbon, nitrogen, phosphorus, and so on, and also the soil pH. For our data analysis, we used mixed effects model, and the blocks are used as random effect. And then in our model we included everything. We included the treatments and the elevation and the interaction, and then we check for overdispersion in our model, and the we estimated the means of the model by using the image package in R, and we also did some extra analysis on isolated effects by using log response ratios. And for community composition structure, we used Hardy ordination to see how community composition differs among the treatments at the low and high elevation. For the post-processing of data, so our team of botanists goes in the field roughly about 3 months, and then they do cover recording by using brown blanket scale, and just to see how plant richness or the composition change from in within 3 months or from 3 months interval. And then During the time of harvest, any trees which is more than 1 centimeter, we measure the BH, and all the plant species were collected per plot into a big mosquito net, and then from there insects were collected in 70% ethanol and then morphotyped at the base. We only collected plants that made up 80% of the biomass of each plot, and then we took the leaves, sample leaves, we took Photoshop, we took photo on a 1 by 1 meter grid, and then from there we use image application to calculate the herbivory, the amount of leaf that is eaten by herbivores. And we also weigh the dry weight, the fresh weight and the dry weight, and all those information was used for the SLA, the specific leaf area, water content, and leaf dry matter content as our traits. So here are the results after a year of succession. So the control, insecticide, weeding plus insecticide, and the weeding. So we can see that in control, it's mostly dominated by Hebs. There's no clear difference between insecticide, although there are saplings growing, especially in control. In the weeding plus insecticide, there are woody plants, like piney woody plants, like the plants in the picture on top right. This is Omalanthus, and if you look at the right below, you can see that there's a mixture of plants growing. It's not a similar type of species growing, but a mixture. So based on in regards to our hypothesis that insects will suppress biomass, plant biomass, more on the lower than the high elevation. So, and also with our fourth or the third hypothesis that insects also facilitate by also suppressing non-woody and then facilitating woody. And in the result in figure A, it seems that insects application of insecticide, removal of insecticide is increasing the non-woody plant biomass. And when insects are removed, non-woody plants are able to accumulate more biomass. And if we look at weeding, it's really decreasing compared to the control. And this is as expected because when we are weeding out non-woody plants, so you would expect the biomass to drop. But this effect becomes more stronger at 1700 because both weeding and the combination with insecticide really reduced non-woody biomass. For woody plant biomass, which is figure B, we can see that insecticide, weeding, and their combination increased by woody biomass compared to the control. And then if we look at 1700, there is no effect of insecticide, but it's weeding and the combination of weeding and insecticide really it was more stronger on the high elevation, so they really increased the woody plant biomass. And on the total biomass, combination of both non-woody and woody insects have strong effect on the lower elevation. So in the absence of insects, plants were able to accumulate more biomass, but this effect disappears at the high elevation at 1700. But at 1700, it's mostly the effect of weeding and weeding plus insecticide. So when we do cross elevational comparison, there is decrease of insecticide at 1700, and there's increase of weeding and weeding plus insecticide at 1700. Okay, in plant diversity, in terms of plant diversity effects of weeding and weeding plus insecticide on non-woody diversity are both site-specific and elevational dependent. So we can see that the non-woody diverse insects doesn't affect non-woody diversity, but weeding plus insecticide really reduce woody plant diversity at 700. Um, but interestingly, but also expected, weeding and weeding plus insecticide increase non-woody diversity. So not only woody plants were able to accumulate, increase their diversity, but it was also woody plants when they were removed, they made space for the recruitment of other non-woody plant species. And if we look at figure B, um, removal of insects is decreases diversity. So in the absence of insects, diversity decreases, and we would expect dominance to increase in the insecticide. So there is effect of insecticide on woody plant diversity at the lower elevation. And if we look at the high elevation, um, there was a strong effect of weeding, and weeding increases diversity at a high elevation, and there was no effect of insects. And if we look at the total diversity, which is figure C, yes, there is evidence that on the total community, insecticide lower the total diversity. So we would expect in the presence of insects, diversity increases. There was no effect of insects on total diversity, but there was strong influence of weeding plus insecticide at a higher elevation. So the interesting results from this, the elevation independent result was on the non-woody diversity. And for the woody plant, it was site specific. Even insects have effect on woody plant diversity. Insects are driving diversity, but the underlying reasons or the factors or what is predicting that is not that clear. So we are working on our soil nutrients analysis, the data, and also we're still working on our insects. So we are categorizing them into predators and uh, you know parasitoids, just to see if there is any biotic and abiotic factor that is contributing to that site-specific effect of insects. So for the community composition structure, we can see that at the lower elevation 700, there is a clear distinction in the community composition of control and insecticide. So for the non-woody, clear distinction between the community composition of insecticide and control. And we can see that it's most dominated by Acon is Ageratum conizoides, which is herbaceous species. The Pipan is the Paspalum paniculatum, Paspalum conjugatum, which are all grass. And then, and this seems to favor the insecticide treatment and the control treatment. And if we look at the woody plants, and this is based on the biomass, we can see that the control and the insecticide clusters, the composition of the two are separated, but we can see weeding plus insecticide are not so distinct from each other. But we can see that they are trying to diverse. So if we allow the succession to continue, maybe they will diverse into separate communities. In the two plots below C and D, we can see there is no clear difference when we look at non-woody biomass. There's no clear difference between control and insecticide, so the community composition of the two treatments are same. Also, the community composition of weeding and weeding plus insecticide are also similar, but then so they have the two main clusters. But when we look at woody plant biomass, there is no difference between insecticide and control. Communities are similar, but there is there is a clear difference which is starting to make sense um, in weeded and WI plots. Because and if we allow the succession process to continue, then both will have distinct communities. Okay, about alien native plants. Uh, it's interesting to see that the alien woody plant biomass, the proportion of alien plants based on biomass decreases in the weeding plus insecticide plots. And then if we look at 1700, weeding plus insecticide increases the alien woody plants. So the increase in alien woody plants um, it means that there is also a decrease in the natives. So natives are benefiting from weeding plus insecticide at the elevation, but not at a higher elevation. And if we look at alien woody plants based on richness, there is a decrease in weeding plus insecticide. So based on richness, native plants are benefiting more from the weeding plus insecticides, the combination of the two treatments. So there is a reciprocal elevational effect. So insecticide decreases. Effect of insecticide on alien woody plant biomass decreases at high elevation, and uh, there is increase of um, insecticide plus weeding on alien woody plants at a high elevation. So in conclusion, this study documented the importance of insects in the initial, secondary succession of tropical forest, and the influence of insects was limited relatively to the less stressful environment at the low, which was uh, which was true for the biomass, and it was also true for the diversity, but elevational dependency effect was evident in biomass, but for diversity, it is site specific. It is site specific effect, and there could be some other predictors or factors contributing to that pattern. And competition between non-woody and woody plants increased with increasing elevation, and we can see that competition is more stronger at higher elevation. And insect removal benefited alien woody plants at less stressful environment, which is at low. And while the interaction of insects and non-woody plants facilitates the increase at high elevation. And despite theoretical and practical importance of succession and regeneration studies, our results often lacked comparable studies. This study was funded by Gacha, uh, given to Professor Wojtek Nowotny, who is my principal supervisor, and my co-supervisor is Dr. Piotr Sefer. And institutions that collaborated in this study is the New Guinea Binatang Research Center in Medang, the Jack Academy of Sciences via the Biologics Center, and University of South Bohemia in Saskatchewan in the Czech Republic. So this is our two teams at Numba, and the one on the right is our team at Hubi. Uh, this is to acknowledge my colleagues and fellow who have assisted me in completing that project well. This is the end, and thank you very much for listening.